Bioinfocongress IV Assistant Professor Dr. Akdes Serin Harmancı Machine Learning for Single Cell Data Analysis. Dr. Akdes Serin Harmancı is an assistant professor at Baylor College of Medicine, Department of Neurosurgery. She was an associate research scientist and a postdoctoral researcher at Yale University School of Medicine, Department of Neurosurgery, before working at Baylor College. During her postdoctoral studies, she and her colleagues discovered epigenetic causes of brain tumors and identified new molecular targets for precision. She has studied the epigenetic and genetic mechanisms leading to the atypical and malignant transformation of brain tumors. Dr. Serin Harmancı completed her undergraduate studies at Sabancı University in Computer Science and Engineering. Then, she finished her PhD in computational biology at the Max Planck Institute of Molecular Genetics in Berlin. The primary focus of the Serin Harmancı Lab has been developing integrative approach to dissect the molecular landscape of brain tumors using single-cell RNA sequencing and spatial transcriptomics data. Currently, they are working on developing new methods for understanding tumor heterogeneity and the immune microenvironment in brain tumors using single-cell RNA sequencing for developing targeted therapies.
Dear Dr. Akdes Serin Harmancı and participants, welcome all of you to our Bioinfocongress IV event. Dr. Serin Harmancı will give us a talk today on machine learning for single-cell data analysis. At the end of the presentation, we will have a question-and-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Serin Harmancı, welcome back. It's so great to have you here.
Thanks a lot, Efekan. Thank you for having me here. And thank you for the great organization. I know you have been working on this for some time, and it's a great organization. Thank you. So I think I can start with my presentation now. Today I will be talking about machine learning for single-cell data analysis. So I will try to give an overview of single-cell technology, and hopefully I will go through some computational challenges in single-cell transcriptomics. I will also talk about past and current research on brain tumors that we have been working on for some time. And I will briefly go over trend topics in computational biology. Let's start with the overview of single-cell technology. You might be aware of this single-cell technology. It's been around since 2013. It was the method of the year in 2013. Since 2013, these technologies become routine when investigating cell-to-cell heterogeneity and transformed many areas of biology and medicine. Here is an overview of the scaling of single-cell RNA-seq experiments. In 2010, like the single-cell technologies were able to sequence 1 or 10 cells, but thanks to microfluidics, we now can sequence around million of cells, and we have a lower sequencing cost, and all these things, microfluidics, low sequencing cost, it empowered single-cell sequencing technology. And now, as I said, like we can sequence millions of cells in an experiment, and this is a huge data, as you can imagine. So it's a data science problem that we have. So this is like a figure that shows the difference between the bulk sequencing and the single-cell sequencing. In bulk sequencing, we have a sample that contains like thousands or millions of cells, and DNA is extracted from all of these nuclei. And then we have the reads coming from all of these cells in the bulk sequencing, and we map all of these reads to the genome. However, in the single-cell sequencing, we try to extract DNA from one single cell. And we amplify the DNA, and then that amplified DNA coming from one single cell is sequenced and mapped to the genome. Here we have, as I said, like a data science problem. Here we have millions of cells coming from one tissue. But on the other hand, the data is noisier compared to the bulk data. It's much more sparse. So we have to treat the data in a way that we can remove all of these noises, and also we have to think about the sparsity of the data. This is a well-known analogy that shows the single-cell transcriptomics. It has been adapted from the Human Cell Atlas. So this really explains it well. In the bulk RNA-seq, it's like putting all the fruits in a blender, and we taste the average of all of the ingredients. In single-cell RNA-seq, we can taste all of these individual fruits. We can understand the detailed composition of the fruits within the fruit salad. So bulk RNA-seq is like a fruit smoothie, and the single-cell RNA-seq is the fruit salad. So single-cell data is much larger, and it's much more sparse. We have lots of zeros in the data, so we have to think about how to get rid of the zero values in the data. There are broad applications of single-cell sequencing in biological and biomedical research. For example, in neurobiology, we can use, it has been used, single-cell sequencing technology has been used broadly. There are lots of consortiums like Human Brain Atlas, Allen Brain Atlas, Human Brain Somaticism Network, and all the other consortiums. They sequence like millions of cells, and they try to understand like different neuron cell types. They would like to see the somaticism in the brain cells, and there are also other applications in immunology. There are like tons of different immune cell types, and it's not just like discrete cell types. We have states, so it's like a spectrum of cell types, and with these technologies we can build trajectories to understand all these different cell types and states. Here we will focus on cancer biology. My research is mostly focused on the cancer biology. And what are the main applications of single-cell sequencing in cancer research? We can resolve intratumor heterogeneity. As you know, in cancer, we have intratumor heterogeneity. We can investigate the clonal evolution in primary tumors. We can understand the invasion and metastasis. We can also profile, we can sequence and understand the circulating tumor cells. We can look at the mutation rates and the main driver mutations happening in the tumor cells. We can understand the resistant evolution to therapy. We can understand the cancer stem cells and cell hierarchies. And also we can study the EMTs, epithelial to mesenchymal transitions, by building trajectories. I said we can understand intratumor heterogeneity, metastasis, plasticity, and so on. But also we can have a better idea about the tumor microenvironment. We can characterize different cell types, different cell type compositions within the tumor microenvironment, and that is very critical in guiding the treatment of tumors. For example, the ratios of CD4 or CD8, T cells have been shown to predict overall survival and different immune responses. With the use of single cell technologies, we can understand the different immune cell type compositions within the sample. And this is like a figure that shows what kind of information we can get from the single cell data. Right now we can zoom in to one site of tumor that is happening in the body, and we can see that within that tissue we have a lot of different cell types. We have the tumor cells, we have the T cells, B cells, dendritic cells, fibroblasts, endothelial cells, T cells, and so on. When we do bulk RNA sequencing, here we see like 3 gene expressions in this plot, in this figure. When we do bulk RNA sequencing, we just take the average of that gene, and we don't look at each of these cell types, but we just take the average of all these cell types. So it just gives an overview. However, when we do the single cell sequencing, we can understand what is that gene's expression within the neoplastic cells, within the T cells, B cells, and all these other vascular cells. Yeah, and that can be used in personalized medicine, because we can understand like what is that single normal cell that transforms into a tumor cell. We can get a better idea about the clonal evolution. We can see how that single cells from the primary tumor form metastasis. And we can also understand the single tumor cells that survived the therapy, and we can potentially like develop targets like drugs for these surviving tumor clones. So understanding the clonal architecture of the tumor is very essential in clinical care, especially for determining treatment-resistant tumor subclones. Yeah, this is another figure that shows the same idea. In the bulk analysis, we get an idea about the average clone, and we develop a drug for that average clone, but we have other clones in the tumor. And if you just look at the average clone, we cannot really remove the tumor cells. If we have the single cell analysis, we can get an idea about all these individual clones, and we can have precision drugs targeting each of these individual clones. We said that like with the single cell technology, we have millions of cells. So it's a data science problem, and we need computational methods and techniques to remove the noise in the data, to interpret the data, and to get some meaning out of this data. This is like a basic overview of the single cell RNA sequencing processing pipeline. We have the patient tumor. It's dissected from the tissue. We have, as I said, like the malignant cells and all these non-tumor cells that is in the tumor microenvironment. And each cell within this tissue is sequenced. We have, after sequencing, we have a lot of reads. Like it's just like ACGT letters coming out from this sequencing machine. And what we do is like we map all these reads to the reference genome, to the human genome, and then we generate single cell count matrices. And these matrices have like genes in the rows and cells in the columns. We have, you can imagine that we have like 20,000 genes in the rows and 1 million of cells in the columns, let's say. And the other issue is that, as I said, like this matrix is full of zeros, so it's zero inflated, and our tools or methods have to take this into account. Because of the dropout, we might not be able to detect the expression of some of the genes. Some people do imputation before doing further downstream analysis. There are also some maybe dead cells that we have to remove. There might be some doublets. There are tons of different tools that are available to do all these analyses. I listed some of those here, like Seurat analysis pipeline is like one of the most popular ones to do all these pre-processing pipelines. There's Scampi, and if you are doing 10x single cell sequencing, 10x company has its own mapping tool, Cell Ranger. But we still lack some algorithms and methods that can properly analyze all these data sets. One of the other main issues is that batch correction, data integration, and dimensionality reduction. We might have data coming from different patients, and we would like to integrate all of this data, but there might be some patient-specific biases that we have to remove. So first of all, we would like to do a dimensionality reduction because we have, as I said, like 20,000 genes, and we cannot really visualize 20,000 genes and 1 million cells. One of the most popular ways of visualizing this data is doing dimensionality reduction. In bulk RNA-seq, we usually do PCA. It's a linear transformation, but in single cell data, PCA doesn't work well. Because of the sparsity of the data, we need a nonlinear transformation. So people started using t-SNE or UMAP. Those are like nonlinear transformation techniques to visualize the data. So we had this 20,000 genes and millions of cells, let's say, and then we can reduce 20,000 gene dimension to 2 dimensions. We have like, this is like a plot of UMAP, dimensionality reduction plot. We can visualize the data with the UMAP nonlinear transformation tool. Here, each dot represents each single cell. And after we reduce the dimension, we cluster the data. And then hopefully each of those clusters are like homogeneous cell types that represents each cell type. Basically, as a summary, dimensionality reduction is needed for visualization, for clustering, for simplifying the complexity of the data, reducing the number of features in the data. And the other issue is the batch effect correction and the data integration. Let's say we have a study where we have drug treatments, like with drug treatment and control, 2 different data sets, and we would like to combine these data sets that is coming out from the single cell sequencing, the mapping platforms, the mapping tools. We would like to combine all these outputs of these gene cell count matrices. However, when we just combine it like in a very simple way, we would see that there's a study bias in the data, where we see that each of these like treatments are clustering within themselves mostly. So we need to have data integration methods that can align those data sets. So there's like different methods that can align those 2 data sets. One of them is the canonical correlation analysis that people use. So this is basically like aligning those 2 data sets in like another dimension. And after aligning the data sets, then we can say what cell types have different proportions in under which treatment. We can also get like cell type specific transcriptional responses to drug treatments after data integration. All of these steps are very crucial, as I said, dimensionality reduction, data integration, and I didn't mention it that much, but like the initial normalization is also quite important. Yeah, so let's move on. And now we will we have the clustering of the data. We reduce the dimension. We have the clusters, but we would like to annotate each of these clusters. Annotation of each of these clusters is a difficult task. People have been using different methods, but the main idea under all these methods is that, like, within each cluster, we expect to see one cell type, and we look at the markers of these cell types, and we annotate each cluster based on the marker genes. But the issue is that it's not always the case. It's sometimes we see heterogeneous profiles within each cluster. And the other issue is identifying tumor cells within this clustering. Again, tumor cells are not always clustered together. They might be heterogeneous. So what we do is we try to utilize the single-cell RNA-seq data and try to infer genomic profiles from the single-cell RNA-seq data. And as you know, like, we see copy number variations in tumors, and if we can infer large-scale copy number variations from this data, then we can have a better idea which cells are tumor. And you know, we have an accumulation of somatic mutations within the tumor cells. So again, inferring mutations from single-cell RNA-seq data might facilitate to identify all these tumor cells. And again, as I mentioned before, so we have two components in cancer single-cell RNA-seq data. We have the tumor cell identification step, and we have non-tumor cell identification step. And people have been using different ways. We can either look at the marker genes and try to annotate each of these clusters based on some known marker genes. We can have correlation-based methods where we have a reference data, and we have the labels of each of these cell types in the reference data, and we correlate our data with this reference data, or we train the data sets and we train known, well-labeled, well-annotated single-cell data sets, and then predict the cell type within our own data. So I will talk about our past and current research on brain tumors that utilizes single-cell RNA-seq. But first, I will go over my postdoc studies where we mainly used bulk RNA-seq or whole exome sequencing technologies. So I will briefly go over my past research on brain tumors. I've been working with the brain tumors for some time now, and we have been focusing on gliomas and meningiomas. Glioblastoma is a lethal disease. Average survival for patients with GBM is 15 months. And unfortunately, survival rates have not changed in over 30 years. And the brain is a therapeutically challenging organ system. We might not be able to take biopsies from the tumor, and operations are not very easy. During my postdoc studies, we were working with the whole exome sequencing data, and we were trying to identify the driver mutations in meningioma. NF2 mutation was known, but we were looking for some other driver mutations in meningioma. And for that, we did whole exome sequencing in around 300 patients. And here in this plot, each bar represents each patient sample, and we have identified driver mutations in TRAF7, AKT1, PIK3CA, KLF4, HACHOK pathway genes, and POLR2A. And we also saw that like all of these mutations are mutually exclusive. Interestingly, this HACHOK pathway genes are targetable by drugs that they have been shown to work. HACHOK pathway targetable drugs have been shown to work in different cancer types. And we have also shown that all of these mutations have correlations with anatomical positions. NF2 mutations are mostly in the tumors that are in the cerebral, and all of these non-NF2 mutations were in the skull-based meningioma. We also looked at genetic and epigenetic causes of progression in meningioma and glioma. We had matched glioma samples that were primary initially and then recurred and progressed later in the later time point. We did integrated epigenetic methylation, RNA-seq mutation analysis, and we have identified some oncogenic pathways that might be playing role in the progression of glioma. And we have done the similar analysis for meningiomas and tried to identify the pathways that might be relevant for the progression of meningioma. So those were like some past studies, my previous studies, and those were, as I said, like mainly based on bulk RNA-seq, whole exome sequencing, and mutation analysis. And we have done, as I said, like integrated analysis to identify the molecular changes within meningioma and glioma, with the hope of finding targets that we can use for developing drugs. My current research interest is understanding the interactions between tumor cells, neurons, and immune microenvironment in glioma, and that we can do perfectly using single-cell RNA-seq data. And this interaction between the tumor cells, immune microenvironment, and neuron cells is very, understanding this interaction is very important, because that might answer what drives the progression of glioma. So we have in-house glioma single-cell RNA-seq data, and currently it's around 500K cells. It's like 500,000 cells that we have. Those cells have been dissected from around 10 different patients. They are like fresh taken out from the operating room and sent to single-cell sequencing right away within a couple of hours. So we are trying to understand this 500,000 cells is coming from different patients. We have this data integration issue. We don't want to have any effects of or any biases of different patients that are not biological effects. We have to remove all these non-biological effects from the data. We have to analyze all of these data sets and understand the intratumor heterogeneity. We have to understand the tumor microenvironment. And instead of annotating each cluster, we would like to annotate each single cell, because especially in brain, we have a high level of heterogeneity. So we need a single cell-based annotation technique. So let's go over each of these points. And first, let's focus on differentiating tumor cells from non-tumor cells. So as I said before, we don't have simultaneous single-cell DNA and RNA sequencing. That's why we need to utilize the single-cell RNA-seq data and integrate the genomic landscape of the tumor cells with the transcriptomic landscape. So in order to infer all of these genomic changes, we use our CMV inference algorithm, Casper, and SNP mutation inference algorithm, Excavator. So, you know, like copy number variations happen in cancer cells, and we have large-scale copy number variations happening in cancer cells. We have large-scale chromosome 22 deletions in meningiomas. We have chromosome 7 and chromosome 10 CNVs, large-scale CNVs happening in IDH wild-type gliomas. And with the read coverage, we can have an idea about the CMVs that are happening in the single-cell RNA-seq data. Very local CMV events might not be very easy to detect using single-cell RNA sequencing, but we can have a good idea about the large-scale copy number variation events. Yeah, so we can also use the variant allele frequencies to better characterize copy number variations, because if we have a deletion, then the variant allele frequency will go up to 100%. If we have amplification of one copy, the variant allele frequency will go around will be in the line of 0.3. But if we have amplification of 2 copies, then we won't be seeing any shift in the variant allele frequencies. So one of the main challenges here is that we have a heterogeneity, and in our tissues, we have a mixture of cells that have CMVs and without CMVs, because in our samples, as I said, we have non-tumor cells too. And we need baseline control samples to correctly identify all these large-scale events. And we can use all these publicly available normal brain atlases like GTEx, Human Cell Atlas, or HubMap. Or we can detect the non-tumor cells within our single cell data and use those cells as a control for our CMV analysis. This is an overview of our CMV inference tool, Casper. So basically, we have the reads coming out from the single cell data. We map those reads to the genome. We generate the normalized gene expression matrix. And then we smooth this expression matrix by applying recursive iterative median filtering. And we smooth the signal. And after smoothing, we apply hidden Markov model to segment the data. It's a well-known technique to segment the signal. And we assign CMV states to each of these regions. And we use variant allele frequency shifts that I mentioned before to correct for our CMV calls. And this is an application of our algorithm in one of the public GBM single cell data sets. Initially, so this matrix that you are seeing on the top is single cell row gene expression matrix. In the columns, we have genes ordered by their chromosomal location. And in the rows, we see each of these single cells, and the color labels that you see on the rows are represents each patient sample. So we have 5 different patient samples here, and each of these patient samples have like hundreds of cells. So initially, that the row gene expression matrix doesn't give any information about the large-scale CMVs. But after we do smoothing and recursive iterative median filtering, we can see that some chromosomes are amplified within some patients. So As I said, like in the columns, we have genes ordered by their chromosomal location. You can see each of these chromosome labels here on the bottom, 1, 2, 3, 4, 5, and like up to 22, chromosome 22. And we see like a high expression signal in patient MGH30, for example. And we have the deletion of chromosome 10 in all of these samples. We see that like MGH29 has deletion of chromosome 15. So basically, using these techniques, we can have an idea about large-scale CMVs within each tumor samples. And hopefully, we can also see some intra-tumor heterogeneities within one patient sample. And so from the smoothed signal, we binarize this data to like either amplified or deleted. And we see like from the signal, we can have another matrix where we have the large scale events marked. If it's deleted, it's blue. If it's amplified, it's red. And we can also visualize the mutually exclusive events within one sample using our tool. So we have an edge in between the CMV events if that occurs like mutually, if it's co-occurrent, and we have a dashed line if it's mutually exclusive. And this is the application of our tool on our in-house GBM single cell data. In GBM, we have chromosome 7 amplifications and chromosome 10 deletions. It's widely known. And by applying our data, we can see that we can detect the tumor cells using CMV calling, using the output of the CMV calling algorithm. We can see where the tumor cells in our clusters. It is nice to see that they are not really homogeneous. There might be different tumor cells, and they might not cluster together. And we can identify which cells are tumor using the CMV calling. But sometimes CMV calling might not be enough because especially in IDH mutant tumors, we don't have CMVs. So we need some other features that might support the evidence of tumor cells. So as I said, in IDH mutant gliomas, we sometimes don't have any large scale CMVs. So we will utilize the mutation calls that we can have from the single cell data. So we applied our algorithm excavator for characterizing the variant impact from the single cell RNA-seq data. Here, the red points are the cells with the mutation, and the blue points are cells without the mutation, and the gray points are the cells without any coverage. So in single cell data, we have a lot of cells, but they usually don't have coverage for all the genes. We have 1 or 2 reads per gene per cell. So here we can see that these clusters that we show here, the red ones, they are tumor cells because they have an accumulation of IDH mutation. And that is an evidence for us for labeling those clusters as tumor. And we have been talking about, until now, we have been talking about the tumor cells, identifying the tumor cells and understanding the intratumor heterogeneity. But we also need to understand the tumor microenvironment, especially in gliomas. Brain infiltration is very high. Brain tumor cell infiltration is very high, and tumor microenvironment changes the tumor cell behaviors, and also vice versa. Tumor cell infiltration changes the tumor microenvironment, and we would need to understand the changes that are happening in the tumor cells. And for understanding the tumor microenvironment, what we did was we have like public data sets, healthy brain data sets that are available like from all these consortium data sets, human alien brain atlases, developing brain atlases, and like human cell atlas, immune cell atlas. We have also publicly available glioma immune cell data sets. So what we did was we trained all of these like data sets with a neural network. We have around 1 million of healthy brain or GBM brain data sets that are well annotated. Like we know which cell is which cell type from these data sets, from these publicly available data sets. So we can use all of these publicly available data sets as a training data set. And what we did was, as I said, like we used all these publicly available data sets as a training data set. And we then predicted the non-tumor cells within our in-house single cell data using all these trained data sets. Yeah, so after putting together all these like methods and steps, we ended up with a pipeline that we call Scrum. And in Scrum, for non-tumor cell annotation, we use the training models of the healthy brain data sets or publicly available well-annotated GBM data sets. And we also make use of our CMB calling algorithm and SNP calling algorithms. And we combine all these annotations in single cell resolution. And we try to see what kind of co-occurrences we see in single cell resolution. So we might see some tumor cells that show like neuron properties or immune properties. So with our Scrum pipeline, we were able to identify neuron-like tumor cells in IDH mutant glioma samples. So within the complex tumor microenvironment, we have detected that there are tumor cells that can adopt partial properties of non-tumor cells to favor tumor cell survival, proliferation, invasion, and metastasis. And like these glioma stem cells that are in the tumor cells that might lead to transdifferentiation into functional non-tumor cells such as neurons. So using our pipeline, as I said, like we have detected neuron-like tumor cells. So these are some of the figures. So our paper is in bioRxiv right now. We are still making some changes into our pipeline, but basically what we show here is that like in the panel of A, B, C, and D, we show that there's a cluster in our single cell data that have copy number variations and IDH mutations, and also express markers of GABAergic neurons and also glioma stem cells. So in order to experimentally validate all these findings, we did PAT-seq. PAT-seq is a very recently developed technology. We can do whole cell recordings and whole cell patch clamp recordings, followed by single cell RNA sequencing. We can get the electrophysiological properties of each single cell using this technique. From the PAT-seq, we have seen that some of the tumor cells have electrophysiological properties of neuron cells. And these findings were specific for the IDH mutant gliomas, and we have not detected this finding in the wild-type mutant, in the IDH wild-type gliomas. And we have also found some immune-like tumor cells, and those might be also relevant for the immunotherapies. Yeah, so if you want to look into the details of our findings, maybe it will be better to go into bioRxiv paper. And next, I would like to just briefly go over the future trend topics in computational biology. So, you know, deep learning is everywhere. We have like ChatGPT and all these like complex deep learning models that we have been talking about recently. And deep learning is also in bioinformatics, in machine learning field. There are tons of different applications of deep learning models in single cell data, especially because, as I said, like single cell data has like millions of cells, and that is a perfect data for deep learning. We have a lot of data points that we try to understand and learn. So deep learning methods have been used widely for single cell RNA-seq data analysis for different purposes, for removing the noise in the data, for imputation of these 0 points, or for cell annotation. It has been widely used. Or transfer learning, it has been used for this concept called transfer learning has been used to map different type of data sets and learn from one, maybe like the from mouse model, we can learn the human model and so on. And another application of deep learning was about predicting the effect of mutation. We have like billions of A's and C's and G's and T's in our genome, and we would like to understand if a mutation has a deleterious effect or not. And people have been doing deep learning to understand if a change in the mutation, if a change in the genome would lead to a deleterious mutation or not. Yeah, and also I was talking mostly about the single cell RNA sequencing. So I said like single cell RNA sequencing is currently in everywhere in all the studies, the number of publications related to single cell sequencing has been going up exponentially. But recently, within the last couple of 2 years, now we have also specially resolved transcriptomics technology. So in this specially resolved transcriptomics technology, we can have an idea of the gene activity, the expression of the gene, close to single cell resolution, and also we know where that gene is located within the tissue. So if you go back to this fruit smoothie analogy, we have the fruit salad. We said bulk RNA sequencing is like a fruit smoothie. We just get an average taste of the ingredients. In single cell RNA-seq, we said we can taste each of these individual pieces of the fruit. And in special technologies, we can rebuild this fruit salad. We know the physical location of each of these fruits in our fruit salad bowl. We know where is like bananas are located in the bowl, where the strawberries are in the bowl, and where the blueberries are in the bowl, and we can reconstruct this this physical location using all these special technologies. And if you are interested in single cell genomics, I wanted to include this. This is like a single cell genomics day. I think that's a very nice workshop that is free. And if you want, you can just register for this workshop. And it's on Friday, April 7th. And I would like to acknowledge all of those, all of our collaborators, like all these computational methods we developed together with Arif Harmanci and also Su Wang, Sha Chan, and Xiaobo Zhao. And for all these experimental validations in the mouse models and providing all these experimental support, I would like to thank Rachel, Benjamin Denin Lab, Akash Patel, Timo Klish, and I would like to, and Malcolm McDonald, and I would like to thank Dr. Rao and Dr. Patel for providing human patient samples that we did single cell sequencing, and all the previous work that has been done at Yale about meningiomas. I would like to thank Dr. Murat Günal lab. And I think that's it. And if you have a computational background, you can email me for any possible postdoc or intern positions. Yeah, hopefully it was useful for you all. And I will be happy to get any questions. I see that there are some questions coming up. Yeah.
Thank you very much for this precious presentation, Dr. Serin Harmancı.
Thanks a lot, Efekan. Thank you for inviting me again, and thank you for the great organization. I think you did a great job, like putting everyone together in a very nice way.
You're welcome. Then I'm starting with the first question. What solution can machine learning offer for dealing with errors and missing data in the identification of variation from single-cell DNA sequencing data?
So if we are only talking about the DNA sequencing, not RNA sequencing, so right now, most of the data that I was talking about was about RNA sequencing. DNA sequencing has been developed also for like for 10 years or 15 years now. But it's not as wide as in single-cell RNA sequencing because the amplification is not that easy. So we have a lot of errors. So that's a great question. Like we have a lot of errors and missing data in the single-cell DNA sequencing data and also in the single-cell RNA-seq data. So what we do specifically for the DNA sequencing data or calling mutations or identification variations, we pool all the cells coming from one patient together so that we have a higher power. It's like I don't know if you are familiar, but like there's this famous GATK pipeline from the Broad Institute that people use for variant calling in whole exome data. So it is kind of a similar idea that we do. We have to pool all the cells together. In this GATK pipeline, they also pool all the samples together to increase the power. We pool all the cells together in the DNA sequencing data, and we call variants from all these pooled data, and then we go back to each cell and genotype each cell based on the mutation calls of the pooled data. I don't know if it makes sense, but to you. But so the other thing is, of course, we might have, if we are focusing on cancer and if we are studying cancer, we will have germline variants, and we are not interested. I mean, some people might be interested in, but mostly we are interested in the somatic mutations, the mutations that are happening specifically in the tumor cells. So what we do is, like, after doing the variant calling, we look at the databases like COSMIC. It's like well annotated tumor mutation databases. So we look if our mutations are also happening in the tumor databases, if they are also there in the tumor databases. And if we are looking for an exact mutation, like an exact change in the DNA at an exact protein location, then that is much more easier. We can be more confident. Yeah, so hopefully that answers.
Thank you very much for your answer. Can you hear me?
Yes.
Thank you very much for your answer.
Thank you.
Our next question is, how can we select cell population for tumor and immune cell crosstalk before and during malignant progression in single-cell transcriptomic?
So that is a great question. So this question is mostly about the cell-cell communication, I think. So first, we need to annotate each of the cells in the single-cell data. We have to have this annotation of the tumor cells, immune cells, and so on. And then the next question that we can ask is, what is the cell type communication that is happening here? And I didn't really go into that question in this talk, but people have been looking at the receptor ligand expressions like there has been databases that show like pairs of receptors and ligands, and after we do the annotations, we can go over all these receptor ligand pairs and expression of these receptors and ligands within different cell types and try to infer the crosstalk between those different cell type populations.
Thank you for your answer. Our next question is, can we use ML in single-cell omics data for general tasks such as cell type recognition, methylation, and histone modification?
Yeah, there has been like tons of different methods for cell type annotation that use machine learning, especially Fabian Theis group from Germany. They have been developing different deep learning methods for single cell data sets. And there has been some methods that combine histone modification data sets to predict expression, or like different combinations of omics data to predict the opening of the chromosome, and some other features that we can predict using these different machine learning methods. And I also would like to say that there is a conference coming up in Istanbul, and you might already know, but I just wanted to remind. It's called Recomp, and it's a computational biology conference. It's a very, very popular and well-known, well-recognized conference, and this year it's happening in Istanbul. And there is going to be like great talks from all over the world. It's going to be an in-person meeting, so if you want, you can attend. Yeah. And there will be a lot of, there will be like all these like PIs, like professors working on single cell mixed data and machine learning methods over there.
Thanks for the information and your answer. Our next question is, how can we use machine learning to quantitatively describe the tumor evolution process and evaluate different possible modes against each other?
So there has been some methods to create this phylogeny tree. They call it the phylogeny tree, the mutation phylogeny tree from the data, from the mutation data. There has been different theories how the mutations evolve, but one of the main theories is like a tree kind of like evolution. So there has been some methods that try to reconstruct this tree from the exome data or from the single cell DNA sequencing data.
Thank you for your answer. Our next question is, sparsity in scRNA-seq data can hinder downstream analysis and is still challenging to model or handle appropriately. So how can we improve?
Yes, that's a great question. Sparsity is a very it's a big challenge in this data analysis. So some people do imputation, so there are lots of 0 points in the data, and it's 0 not because it's not expressed, but it's because it cannot be detected. It's like a technological artifact that we have. So we can do imputation. There are like different tools that does imputation. We can predict the expression of that gene at that cell by taking information from all the other cells and genes and impute. And most of the models, the underlying models in all these models, like in these tools, is that it's a 0 inflated data. So the underlying statistics is based on this 0 inflated distribution of the data. Yeah.
Thank you for your answer. Our next question is coming from YouTube. What are the challenges to integrated analysis of multimodal single-cell omics data using machine learning methods?
Yeah, thanks a lot for the great questions. So that's another topic. It has been studied recently a lot. This integrated analysis of multimodal single-cell data. because we have proteomics data and we have like single cell attack-seq. That is like the next common data that we have. We have single cell attack-seq and we have the single cell RNA-seq data. And what we want to do is like we want to integrate single cell attack-seq and single cell RNA-seq. And we can integrate these data sets in gene level. We can predict the gene level activity of cells for the single cell attack-seq, and then we can use some integration methods to integrate the single cell attack-seq with the single cell RNA-seq data. One of the ways can be like this transfer learning based methods, and also all these like the correlation, canonical correlation analysis that I was mentioning, that we try to align those two data sets into the same space. That is another thing that we can do. And there are different tools available, like Seurat pipeline is also a nice tool. Yeah.
Thank you for your answer.
Yeah.
Moving on, machine learning has some challenges, such as single-cell omics data quality control, noise processing, and batch effect. How can we solve these issues?
Yeah, again, we have we know all of these issues exist in the data, so we have to be careful when we are doing the analysis. We should be aware of what kind of problems that we can have in the data, so that we don't make any non-biological conclusions out of this data. So we have to remove the batch effect in order to properly align the data sets coming from different studies. And we have to remove the noise. As I said, like we can either do imputation, and quality control is also important, because we might have a lot of barcodes in the single cell data that don't contain any cells. So we have to remove all these barcodes within the data. Yeah. But there are like lots of tools for all of these things. People have been working on this for a long time. Yeah.
Thank you for your answer. Our next question is coming from YouTube. Do you think the learning difficulty is a disadvantage for researchers because of the exponential number of approaches for dealing with single-cell genomic data? With over 800 published tools for scRNA-seq analysis, most of which are based on ML approaches.
Yeah, that's a great question. So that is an issue that I agree. There has been a publication like last year from, again, Taste Group, that they said like there are more than 1,000 tools available for different single cell analysis purposes. I agree that there is like a lot of tools for different problems in single cell RNA-seq, but the good thing is like it is still improving, so it's building on top of each other. So the reason why we had to develop our own pipeline to analyze our in-house data is because the existing methods were not effective in analyzing our own data, because it was not brain cancer specific. The tools that are available were not brain cancer specific. They were not really asking the questions that we were interested in. So we had to come up with another tool, even though there exists like tons of different other tools. So there is still a need for new methods and tools, because everything is like, every problem has its own questions, and like answering those questions requires sometimes developing new tools. Yeah.
Thank you for your answer. Our next question is coming from YouTube again. How can to analyze the single-cell omics data with the increasingly larger gene expression matrix contained many zero items and data with multiple types of noise generated by different sources?
Yeah. So again, we have to be careful about all these issues, and so we can either follow some standard analysis pipelines that are available to like remove the batch effect and noise and things like that, and also look at the data by eye a lot, like going over all these like plots and trying to make sense out of the data by really being together with the data. Yeah.
Thank you for your answer. Our next question is, while differential expression analysis, gene expression may largely vary within a group or be expressed by a percentage of cells at the single cell level. How can we determine and evaluate whether these effects were representative of a phenotypic shift or a subset of cells which change during cancer progression?
Yeah, so most of the standard tools, so that's a great question. So usually, like, as I said, like the typical way of analyzing the data is reducing the dimension, clustering, and then looking at the changes in expression within each cluster among different conditions. So, but these pipelines have the assumption that all these clusters are like really homogeneous. But in our case, in the brain data, we have seen that it was not the case. So we, instead of doing cluster-based analysis, we did single cell-based annotation with the hope of having hopefully more homogeneous like cell groups. So that is an issue. If you start from the cluster-based analysis, if your data is really heterogeneous, like brain cancer, then you might end up with a lot of variability within one cluster. So it might not be very easy to evaluate the expression changes. And you might not really know if it's because of any artifact or if it's real. Yeah, so that's an issue.
Thank you for your answer. Our next question is coming from YouTube. Which features of HCVATR can be distinguished from other available methods?
Yeah, so we developed this tool because in our in-house data, we would like to identify the tumor cells. So the input is the single cell RNA-seq reads. So we pull all the reads coming from all the cells, and then we call mutations, and then we go back to each cell and genotype each cell by each mutation. And so at the end, we what we do is like we map the mutations to the UMAP or the reduced dimension, and then we do shuffling, coverage-aware shuffling to assess the density of the variant within each cluster, so that we can have an idea whether that cluster is tumor or not. And we also look at the cosmic annotations, and we look at rare mutations. We filter out all these like mutations. We don't look into common SNPs. We focus on the mutations that are reported in cosmic database. And yeah, that's it basically.
Thank you for your answer. Our next question is, today we have a lot of approaches in data science, but why could not we develop common, powerful, and reliable analytical methods and computational tools?
So that's a great question. So you know, like in like there are like deep learning tools applied on different data sets, like images currently. Like we can try to distinguish if an image is a cat or a dog and other like another object. So those questions are, I think, easier compared to the biology, because biology is like really complex. We don't really know what it is. We don't even like, even the training data sets that we have with the labels, they may not be correct because there's no correct labeling, because we don't maybe have discrete cell types. We might have a cell type spectrum. It can be like not a discrete cell type, but like a cell state. So even having we don't even have well annotated training data sets. So I think biology is much more complex than the other data science problems. that we have for images, for other like maybe language models that we try to have in the ChatGPT or things like that. So we don't have the like the training data, well-labeled training data. The biology is so complex, and everything is like all these genes are interacting with each other, and yeah, so it's not very easy to get that information out of the models.
Thank you for your answer. And our last question is, which advices Dr. Serin Harmancı may give for students studying in scientific fields?
So it's like currently it's great. Like I'm also learning. We are continuing on learning. If we are not learning, then we will be out of date. So that question applies to me also. So I'm trying to learn, for example, all these deep learning methods and techniques because they did not exist when I was studying. So the good thing is there's a lot of resources available online, and like all these courses are available online. And for coding, all these AI tools are a great start. If you ask like how you can write this in Python or how you can write this in R into all these AI tools, that gives you a good starting point. But from my experience, I think it's great that we have a lot of resources available. We have all these Coursera's and YouTube videos of all these like professors. It's great to have all these things, but I think it gets more useful if you have like a problem in hand. Let's say if you want to solve, you have to start with a problem. You have a data and you want to solve. You have a problem that you want to solve using this data, and then you start learning, because otherwise you just listen something and then you forget about it. But if you are trying to solve a problem, then you actively try to understand how you are going to resolve it. So that's how I do it, basically. That's that's how I try to learn myself. Yeah.
Thanks for the great advices.
Thank you.
On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this informative presentation.
Thank you. Thanks a lot. And also, I would like to say, like, if there are like from the earthquakes and all these disasters, like for the victims, I would be happy to like be in contact, like also give advices about any computational problems that they might have, like. And also anybody like who is interested in this topic, you can write me, and maybe we might have some positions available. Thank you. Thanks a lot.
Thank you so much. I wish you good and healthy days.